All right, last parts of this chapter, but first, let's do a little review here. We have innate immune cells are relatively non-specific and recognize invasions by recognizing what we call conserved, which means common or related, microbial molecules. That's our patterns. Which of the following is most likely to be recognized by an innate immune cell? So, is it A, cholesterol, B, influenza HA surface protein, C, staphylococcus protein A, D, peptidoglycan, or E, T4 bacteriophage tail fiber? Think about this deeply. Think about this part of it and this part of it and look at those answers. Pause the video, think about that. All right, so key points. Innate immune cells are relatively non-specific and recognize conserved patterns, so things that lots of microbes have. Okay, cholesterol, that is something that we have in our membranes. That is probably bad if our immune system recognizes that. So right off the bat, cholesterol's out. Influenza HA surface protein, staphylococcus protein A, and T4 bacteriophage tail fibers are all very specific things to recognize. That does not fit with what we know about the innate immune system. So that leaves peptidoglycan, which is a very common microbial molecule. So that is more likely what the innate immune system would be recognizing. Okay, in 15.6, we're talking about fever. We're going to talk about what fever does and what its causes are. There's pros to it, there's cons to it. So, normal body temperature, as we know, somewhere between 36 and 38 C. Fever is anything above 38 C, so 104 Fahrenheit. Fever is not caused directly by microbes or being sick. What fever is caused by, actually, is by the little thermostat that is in your brain called the hypothalamus. This regulates your internal body temperature. It does this in response to many things, like having an infection or something like that. We'll talk about why it does it in a sec. But the hypothalamus can turn up the thermostat and basically bring your body temperature up. It can do this by what we call vasoconstriction, which is tightening of the blood vessels. When it's cold, it actually cuts off circulation to your extremities, right, to keep your core body temperature high. Or it can modulate it the other way, vasodilation, relaxing of the blood vessels that releases heat to various parts of the body and heats them up. That would be what would cause a body fever to occur. So why might this happen? Well, there are substances we call pyrogens, so pyro means fire, right? So things that bring on heat that can cause fever. There are external pyrogens that will trigger the hypothalamus, like bacterial toxins. Those will tell the hypothalamus to turn the heat up. There are other molecules that bacteria can secrete that can do this. There are also our own molecules that can do this. Interferons and cytokines, which we've talked about previously, get released during inflammation, right? Those are going to turn up the thermostat. So they're going to be in response generally to a bacterial infection, that those are going to be made. So what's the advantage of fever? Well, fevers aren't very fun, right? It feels terrible to have this high fever. But what fever does is it puts pathogens out of their comfort zone, right? It raises the temperature up to a point where they grow worse, actually. Most pathogens grow at 37 very well. So if we raise the temperature above that, they're going to start to slow down their growth, and it's going to enhance the immune response. So we slow the bacterial growth and we crank up the immune system. That is going to help us respond to an infection. So having a low fever is actually good if you have an infection. And you shouldn't try to reduce your fever because it is actually helping you fight off the microbes. The problem is, and the disadvantage is, that Patients are very uncomfortable when this happens, so they feel bad, so they want you to do something, right? But if your fever gets too high, very high, like 107.6 Fahrenheit, it actually starts to cook and denature the proteins in the brain. This is where the term fever dream comes from. When you have a very high fever, you often have very weird visions and dreams and things like that. That is because your brain is cooking. So this high of a fever is actually very bad. And when it gets this high, above 107, we really have to start to try and cool people down. So you put them in ice baths, things like this, to deal with that, right? Because that is becoming damaging to the body. Low fever, not a problem. Our body can handle that. It's going to help destroy the microbes faster than our body. Okay, that's fever. Our final bit is the complement system. Complement is a bit difficult to understand. I think the best way to talk about it is to explain the name. So, complement is a series of 20 proteins in the blood that basically help deal with infection. And it was named complement because when people were first discovering, what's in blood and things, they found that these proteins could help complement or enhance antibodies' effectiveness. So they knew antibodies worked, but complement helps increase the effectiveness of antibodies. Complement is technically non-specific, so it is grouped in with the innate system, but it brings in the adaptive system to help as well in some cases. And it's very critical in helping prevent blood infections. Complements actually kind of triggers this cascade of proteins. So one protein activates another, which activates another, so on and so forth down the line. And it can lead to several outcomes. One are what we call membrane attack complexes, or MACs. These punch holes in bacterial membranes that cause the cytoplasm to leak out, killing the cell. They can also attract white blood cells, which can both bring in phagocytes, but also T cells and things like that. And they can facilitate phagocytosis through opsonization, bringing in those antibodies to create little handles for the phagocytes to grab onto. Complement is made in the liver. And if we think all the way back to the beginning of this chapter, we talked about Brad, who died from his infection, whereas the little girl didn't. Brad was an alcoholic, so his liver was not functioning very well, so he didn't make as much complement as the little girl did. There are 3 primary ways complement's activated. I'm going to mention them. They're not super critical to understand. This is getting a little too detailed for us, but the classical system relies on antibodies and the adaptive immune system, so it's kind of circular here. It's working with antibodies to activate these responses. There's an alternative pathway that doesn't need antibodies, and then there's this lectin-based one, which uses mannose binding lectin in response to cytokines released from macrophages. So these, it's not critical to know the difference here, but the goal here is to insert proteins to make little holes in microbial membranes and destroy the cell. So there are various ways this can happen, but I want to show you what we call the cascade here. So like in the classical pathway, we have an antibody that's attached to a bacteria, and that activates the C1 protein, which then activates C2, and it cleaves in half, and C4 cleaves in half, and then they kind of fuse together and create this C3, which then splits in half, which then can lead to opsonization, where phagocytosis happens, or it can lead to a further cascade of C6, 7, 8, and 9, which all go and punch a hole in the bacterial membrane. Do I need you to fully understand this? No. What I want you to see is that there's a whole signaling cascade here that ends up leading to this outcome or this outcome from this. The lectin pathway is slightly different, but ends in the same result. Same with this alternative pathway. They work at different speeds. Alternative is very quick, so it's activated mainly by the innate immune system. This can take time because the antibodies have to be made, and this one's kind of intermediate here. This is majorly, this one, the alternative, is majorly triggered by LPS, that part on the outside of gram-negative cells. That's something, right? But why does it matter? Well, here's a case history to show us why. We have Derek, 16 years old, he's really good at chess, and he got rushed to the ER, and they found him kind of confused, disoriented. So his parents said that he had complained of a sore throat a few days earlier. Over the last 24 hours, he had high fever and then severe headache. So he also has stiff neck and red spots called petechiae all over his body. The stiff neck, the fever, the headache, all of that points towards meningitis, which we've heard a billion times now in this course. So they do that lumbar puncture, the spinal tap, and they find that his cerebrospinal fluid is full of white blood cells. So he has meningitis. They send it to the lab, and they confirm that he has gram-negative diplococci in there. And they do a rapid test and identify Neisseria meningitidis, which is a common cause of meningitis. So the doctor, when he looks at his chart, is not surprised because this is Derek's third time with meningitis, which is actually kind of uncommon for children. They generally don't get meningitis 3 times. The other times he was 9 and 13. So pretty frequently he gets it every couple of years. After this third time, they start to suspect something is wrong. Particularly, maybe he has a defect in part of his innate immune system. And they look particularly at the complement components. The lab confirms that his blood is deficient in the C8 complement factor. So that is this one down here. It's part of the complex that allows these membrane attack complexes to form. So he doesn't have this system to help destroy bacteria. So his body recognizes them, and the adaptive system will kick on, but it doesn't have these initial quick mechanisms to start dealing with the infection. All right, so that's complement. It's another one of those kind of chemical components. 20 proteins naturally present in blood serum, and it activates this cascade that leads to pores being punched in the membrane of bacteria. All right, that is it for the innate immune system. In chapter 16, we'll talk all about the other side of the immune system, the adaptive immune system.